Great. Thank you so much. My name is Mahesh Narayanan. I'm the president and CEO of PepVax. Thank you, Precision Evolution Global, Sam, and the rest of the team for the opportunity to let me present here today at the virtual Precision and Drug Discovery Summit. Before I start, I would like to tell you a little bit more about myself. My background is in bioengineering with a master's in biotechnology from the University of Pennsylvania, where I had the chance to work with two brilliant scientists, Dr. Carl June, whose lab came up with some of the first CAR Ts, and Dr. Dave Weiner, whose lab primarily works on DNA vaccines. And I had a chance to work with both viral and DNA vectors and saw a lot of synergy between the two technologies. However, I realized the difficulty in designing and manufacturing and administering viral vectors, which certainly can be addressed by DNA vectors, but also saw that DNA itself has quite a lot of problems, especially when it comes to transfecting as well as viruses can. And so there still is a dichotomy between the two technologies at that time. And so in 2013, we formed PepVax, which is really a play on peptides and vaccines and how we really envision the future of these two fields. And we developed a novel DNA-based platform called SMARTid. It's a completely synthetic DNA platform that can really replace viruses as a drug delivery and development platform. And our technology takes a very simplified method to delivery, and our Trojan horse approach, and I'll get into what I mean by that a little later, is really can be used to manufacture any type of required cell therapies, gene therapies, and immunotherapies, especially for diseases like cancers, infectious diseases like the one that we're having right now, the SARS-CoV-2 pandemic, and genetic diseases. And we plan to do all of this directly inside the patient. We like to call this the intrinsic approach, although a more traditional term would be the in situ approach. As I move closer to precision medicine, I realized that we were creating these amazing drugs, but we're primarily creating them for smaller and smaller populations, and that obviously caused more problems as we delve deeper into the technology. And so while cell therapies, gene therapies, and immunotherapies have shown amazing results in various disease cases, they've also had a lot of issues, especially with long production timelines, in some cases very high toxicity that can often kill the patient, and in other cases very low efficacy, and most importantly, they have very, very narrow applicability on the cases that they could be used for. However, our technology plans to address all these problems, and one of the biggest ways we plan to do that is by moving many of the engineering and quote-unquote manufacturing directly inside the patient. And this has a potential for it to be much safer and a lot more effective, and can also be used in many other diseases, especially with cancers, influenza, and even cases like diabetes. So our goal was really to create a whole new class of biologics. And I'm sure many of you are quite familiar with these two terms, allogeneic and autologous. But just as an overview, allogeneic just really means generic or off-the-shelf technologies, and some of the existing ones include allogeneic CAR Ts, checkpoint inhibitors, and insulin. But the biggest problem with allogeneic has always been that it doesn't work the same way or as well for everyone, and they have a lot more side effects than actually treating the treatment themselves, the disease themselves. When it comes to autologous, these are a lot more precise, a lot more targeted, because they're using the patient's own cells to address the disease. And these, most traditionally we can look at as a traditional CAR Ts using adenoviral vectors. But as I mentioned before, these have very long production timelines and potential cytotoxicity as well. So our goal really is to bridge this gap between allogeneic and autologous using this DNA vector. And so we're able to get the speed and the efficiency of allogeneic vectors, but also the precision and the efficacy of autologous approaches. And that's really the goal behind our SMARTid platform. So since one of the things I wanted me to address is how we plan to look at infectious diseases and what we're planning to do with SMARTid, I would like to talk a little bit more about what our approach has been towards COVID-19 and how we're looking to address the situation. But before we do that, just a little bit of background on where we are as an industry. Vaccines, certainly have evolved drastically since the 20th century, but we have yet to successfully create any type of vaccines for coronaviruses. Not for the lack of trying. With 100 times more deaths caused by SARS-CoV-2 than SARS-1 or even MERS, there's an understandable rush to develop a vaccine, and there's about 115 vaccine candidates either in preclinical or clinical stage at this time. These vaccines and candidates fall within 9 different types of approaches. I won't go into all of them, but I do want to point out the most common, which are annotated viruses, inactivated viruses, DNA, and RNA. And the differences in the technology and the applications for the technology are certainly depending on the platform themselves. But this could also depend on the population subtypes as children, elderly, immunocompromised patients, and pregnant women. And so of these, I would like to focus on some of the few more touted of these advancements that received immense public and philanthropic support. The first really would be, I think, Moderna's mRNA-based vaccine, Inovio's DNA-based vaccine, and AstraZeneca's adenovirus vaccine. And it must be stated that none of these 3 approaches have been successful towards or to be used as a vaccine in the past, much less having had much success as a therapeutic in any other disease either at this time. So there's a lot of optimism and there's a lot of hope with these approaches, but there's not as much data to support that any of them would be able to provide the 100% protection we're looking for in humans. And while each of these vaccines might go about it differently, they all, in essence, express some type of antigen which trigger an immune response through cell activation. And one of the problems certainly has been the overstimulation of the immune reaction, which can lead to cytokine storms and possibly exacerbate the lung disease, either directly or as a result of antibody-dependent enhancement, which can cause patient's death on its own. So without the sterile immunity of an effective vaccine, we have to assume that the virus will be around for many years. And as patients can be asymptomatic, it would be very difficult to isolate and quarantine those affected. And so everyone will continue to be at risk of an infection. And so this is really the pitch towards antivirals, which include neuroimmunidase inhibitors or monoclonal antibodies, and even newer approaches that have worked for other disease types, like chimeric antigen receptor T cells, or CAR Ts for short, and others I mentioned, microRNA already. They need to be given a little bit more focus as we move forward in infectious diseases, and this applies for both viral and bacterial. And unlike vaccines, new antiviral approaches can be cross-reactive and address more than just one virus. And so, antivirals can be developed ahead of time, in advance of potential pandemics, and can be stockpiled to be used early on in pandemics, which I think has been one of the biggest shortfalls in this current pandemic that we've had, and even in past pandemics. So what is our technology and how does it come into play into this whole infectious diseases category? The SMARTid platform is based on a proprietary circular DNA sequence that has 3 distinct components that make it unique from other plasmids. The first is our nuclear localization, and this is probably the most important one that I want to point out, which really is a sequence that was discovered by my team, and it is able to enable our plasmid, once it gets into the cell, to enter the nucleus up to 90% of the time, which is about 3 to 5 times higher than traditional DNA plasmids. And so we're able to get the transfection levels of viruses now using DNA, which was one of the biggest problems that I saw when I was working on both viral and DNA vectors in the past. And this is the key, to really be using this type of plasmid, DNA plasmid and non-viral vector mechanism to replace our viral mechanisms that are currently used in cell therapies and gene therapies. The second is our protein signaling, which can express any of the drug that needs to be made, either DNA, RNA, or full-length protein, all directly inside the cell. And in essence, we can hijack the transcription and translation mechanism of the cell, whatever cell we're targeting, and we can enhance the expression, especially if it's a protein, for both intracellular, transmembrane, and extracellular aspects. And so, the third aspect is our inbuilt adjuvant, which enables a very strong immune activation. And obviously, this is very important for immunotherapies, might not be as important for cell and gene therapies on their own. But all of these are cassettes, so they can be added or taken out depending on the drug and that's in development or the disease type. The last component here, which I actually have numbered number one, actually, is the payload themselves. And these are traditionally coming from our university collaborators or industry partners, and are the actual genetic sequence of the therapeutics that we want to deliver or be expressed. But for COVID-19, my team has designed our own anti-SARS-CoV-2 drug that can actually be added to our plasmid, and I'll go into that in just a couple slides. And one other thing before I go into the actual design for SARS-CoV-2 is the delivery mechanism, because we have designed the SMARTid platform as a platform. And so we really needed to be delivered in various capabilities. And so we've created two different capabilities, one for systemic or general expression, and these are usually done intramuscularly, and these are used for either simple expression of proteins or RNA, or even expression-induced immune responses if it's being used for immunotherapies. And the second way we've used this is for targeted delivery, and these are obviously for very specific expression in very specific cells. And here we've done intravenous injections using nanoparticles or liposomes to encapsulate our plasmid, and then we've attached them with cell-penetrating peptides and targeted peptides so that they can only be expressed in certain cells that we want them to be expressed in, and doesn't go systemically into random cells. So these are two different mechanisms that we have used, and for SARS-CoV-2, we've actually used a targeted mechanism. And so let me give you an example of how we're using this in COVID-19. Right now, T lymphocytes for CAR-Ts have to be removed from the patient, engineered into CAR-Ts outside the patient, have them grow in petri dishes, really, and then eventually administer back into the patient. And this process is quite painful and very time-consuming and very complicated. And it can take anywhere between 3 to 4 weeks in some cases. So it's a very long process. However, for COVID-19, what we've done is created an anti-SARS-CoV-2 chimeric antigen receptor and created a DNA sequence for that receptor, added it to our SMARTid plasmid, encapsulated it in liposomes, and delivered it intravenously in or near lymph nodes. And we've done this primarily only in mice. We're not yet in patients. But we also have bound these liposomes with specific peptides that can target CD8 cells, CD8 positive cells only. And so the liposomes, once the peptides bind to these cells, integrate directly into the cell membrane, deliver the plasmid, and from there, our nuclear localization takes over, gets the plasmid into the nucleus, and then we're able to convert that receptor into a protein as a transmembrane protein. And this surface receptor targets the SARS-CoV-2 spike protein, and now these cells can proliferate throughout the body, identify any of the COVID-19 disease cells or the virus themselves, and destroy them. And one other aspect that we've added here is that our target that we've discovered is in a very highly conserved region of the spike protein. And we've seen this region in pretty much all the other coronaviruses in the past. And so we anticipate that if you're able to create this treatment successfully, that it can actually be used not only in the current pandemic, but in any potential future outbreaks of coronaviruses as well, not just SARS-CoV-2. So we feel there's a lot of potential for this type of treatment, which is one of the reasons we want to advocate moving towards these type of treatments rather than just vaccines. But unfortunately, our COVID-19 studies are still ongoing, and we've not yet published any data on that yet. And so I'm unable to share that with you. But in lieu of that, we do have, I want to show some data on a very similar mechanism of expression that we actually did in triple-negative breast cancer. So we did the exact same process, but rather than SARS-CoV-2, we actually created a receptor against MAGE-A. MAGE-A is a tumor-associated antigen that's present on triple-negative breast cancer cells. And so we were able to create these receptors against that protein, and did the exact same process in lymph node of mice. And we were able to generate CAR Ts against this MAGE-A protein within 72 hours, instead of the 3 to 4 weeks that traditional autologous development methods take. And we were able to get endogenous level expressions, not just slight expressions, which reflected on the number of different types of CAR Ts we were able to develop. So that's one of the advantages of this is we actually were able to create multiple allotypes of the same MAGE-A receptor, and we were able to create multiple different types of CAR Ts using the splicing mechanism that we built in to that particular plasmid. And so we were able to get multiple expression of different types of CARs, which can address the MAGE-A protein. So in case there are mutations, other populations of CAR Ts can continue attacking the cancer cells rather than being disabled by the mutation. And obviously, we also wanted to make sure that those CAR Ts can, in fact, attack the cancer cells. And so we moved on to another animal study here, where we took the lymphocytes that we made in the previous study, added them to immunocompromised mice that were growing triple-negative breast cancer cells, so tumor cells on them, and we were able to do an adaptive transfer to show that the T cells can, in fact, target the triple-negative breast cancer cells. And what we saw was a 45% slowdown in tumor growth and about an 80% increase in lifespan of these mice compared to an untreated group, as well as the current standard of care, which was doxorubicin, a chemotherapy agent that we used that's primarily used in triple-negative breast cancer at this time. So, and here's a visual representation of what that looks like. As you can see, in the top is a positive control, which certainly was the doxorubicin and had a tremendous growth by day 18, whereas our PepVax CAR Ts, we were actually able to show much slower growth in these particular set of mice. So this is the same data from the previous study, just a more visual representation of that data. So what are some of the advantages to this type of approach versus other approaches and platforms that are currently available? So compared to viral delivery, as I mentioned, viruses are just really hard to make and hard to manufacture. And the biggest problem that we've seen with viruses, especially AAVs, is that you cannot use them multiple times. We often see that usually in the second or third time of delivery into humans, the patients start to develop neutralizing antibodies. And so that is one advantage here, is that our plasmid is completely non-immunogenic. And so we have not seen any type of neutralizing antibodies using our vectors. And compared to mRNA, which we need much lower delivery, because we can actually, as I mentioned before, hijack the manufacturing process and create multiple copies of the mRNA rather than having just one copy of mRNA per plasmid. So one plasmid can actually make multiple copies of mRNA either in the cytoplasm. We have one mechanism that can do that just in cytoplasm, another one that can actually do that in the nucleus as well. And compared to other DNA vectors, we can get much better expression because of that nuclear localization that our DNA has versus other plasmids. So this platform provides the most versatility among all the other platforms and can really be used, in essence, to replace many of these other technologies that are currently being used as platforms, either by one company or multiple companies. And I just mentioned, many of our competitor platforms, but I do want to say that we currently have 3 patents pending on the composition, utility, and method of manufacturing. And we actually have 2 more patents going out later this year. So we should have about 5 patents by the end of the year covering the entire mechanism. Lastly, I just want to give a quick introduction to my team. We are 4 key individuals. Me and my co-founder, Dr. Anton Dormer. We are the co-inventors and the co-founders of PepVax. And Dr. Daniel Achinko is our lead researcher for in vitro studies, and Mr. Elton Norman is our chief financial officer. And we've all been as a team together since 2013. So in as final words, what I do want to say is, given the virulence and severity of SARS-CoV-2, I think it's imperative that we consider these factors in future outbreaks to use antivirals to slow down early local transmissions, especially in key epicenters like we saw with New York City. And this could even provide partial immunity enough to move us towards herd immunity sooner rather than later. And immediate attention needs to be given to antiviral approaches while vaccines are being evaluated. And there needs to be a little bit more coordinated effort, both national and international, to make sure antivirals are available for all populations, even if vaccines might have limited access, either based on populations or countries. So I will leave it at that, and I'm happy to answer any questions.
Thank you, Mahesh.
Thank you.
We do have some questions for you. The first one comes from Shihong Yan. He wants to know, what's the efficiency of the DNA getting into cells in general? And also, what is the efficiency for the targeted delivery getting into T cells?
Sorry, so the first is the efficiency in
getting into cells in general, and then the efficiency of the targeted delivery to the T cells.
Sure. So our efficiency right now, we are seeing, I would say, the same level of efficiency as AAVs. So we are able to get it to in about 75. We've seen as low as 75 and high as 95 efficiency for our systemic delivery, especially when we are looking at muscle cells. For targeted delivery, we're getting anywhere between 65 to 85% efficiency. So it's still very, very high efficiency. And we're creating our own proprietary liposomes as well that are designed specifically to make our delivery even more efficient. And we are working with multiple companies around that. And one of them is actually working on a way to improve crossover into the blood-brain barrier, so we can address more CNS type diseases. So right now we're still very early stage. We're still working through the various aspects of efficiency in different cell types and disease types. But right now we've seen very high efficiency, especially when it comes to cancers.
Okay, and a follow-on question: how long can the T cells maintain the expression, and how many injections or how strong of a dose is needed in order to get an efficacious response?
Sure, and I do want to say that we've only done this in mice, so all these concentrations and all these efficiencies are in mice only. We don't have any human data yet. But when it comes to mice expression and even in pigs, I would say we've seen about 4 to 5 weeks of expression, transient expression of a CAR, and this is specifically just I'm referring to CAR here. So. Protein expression is actually quite equivalent to an AAV. So as long as it's there, it can express as long as it wants. But what really happens is the proliferation kind of lowers the concentration over time. But for an endogenous level expression and and a transient expression, I would say we've seen up to 4 to 5 weeks of expression in T cells or CARs. And when it comes to, sorry, what was the second aspect there? The concentration.
How many injections or how strong a dose would be needed?
Yeah, so right now, and this is actually, I think, the advantage here and how we can really use this for as a replacement for AAVs. Our concentrations, we actually saw overexpression of CARs at 25 micrograms. So that's a significantly low concentration compared to, when you look at DNA vaccines where we use 300 to 400 micrograms. And even if in AAVs, we can usually see around 50 to 75. So they're about half up to a third of what we need when it comes to AAVs or other DNA vectors. And this is, we've done this in about 6 different cell types. We're still testing out quite a few more. Obviously, COVID-19, it's a common theme. It's stopped many of our testing, and our focus has primarily been just on COVID-19 at this time and moving that into human trials as soon as we can. But we are looking to expand that and look at the doses as we move forward in humans. But right now, we're looking at ideal doses anywhere between 15 to 25 micrograms in mice. So you have to calculate how that would be in humans.
Okay. And Shihong had one more question. That is there potential liability of genome integration?
Yeah, so ours is a completely non-integrative vector, but it does have the potential to, and this is with pretty much any vector, some of them might integrate, but so far we've seen, very little or negligible integration in any of the cell studies that we've done. But we can also do a full integration as well with CRISPR, Cas9, Cas12. So there's a potential there if we do want to integrate, but right now it's a completely non-integrative vector, which is why we can do more of a transient expression versus a permanent expression. But I do want to say that we are currently working on a mechanism to do an on-off switch, where we can create, or induce an expression based on a molecule, and then as the molecule disappears, the expression goes away. And that is something that we're working on right now. I can't say much more about it at this time, but that is pretty much our next generation of plasmids that really can be, triggered very minutely based on concentrations of small molecules to turn on and off the expression if it integrates and if it needs to be integrated.
Okay. Now we have one, this is a two-part question. We learned earlier today about hypoxic cells in tumors. Is it possible that the 45% reduction in tumor size is limited because of existence of heterogeneous tumor cells with different antigenic surfaces because of hypoxia? What if you administered 2 CAR T cell lines, a second one key to the core internal cells in the tumor?
Absolutely, and as I mentioned in the slides before, we actually had about 20 different cell lines of CARs that we used in that study. We only tested 2 or 3 of them in the CAR side, and one of them from a systemic delivery side. But I agree. I think that there's a potential for it to be even higher if you're able to target it, specifically to internal and external cells. But that is not something we've done at this time. And our business model has really always been around licensing this platform to our university partners and to other industry partners. So we haven't really done too many of our own studies yet. We're still waiting for a lot of our partners to finish up their studies. And the triple-negative breast cancer, that was actually done using a partner, one of our university partners. And so, we have thought of looking into it, but at this time we don't really have much data, and we haven't really done something that way where we're targeting internal and external. We've just really done splicing mechanisms of expressing multiple different types of CAR Ts against that surface protein. but we haven't really looked at anything past that yet.
Okay. Well, that's all the questions you have right now, Mahesh. Thank you very much for your presentation.
Absolutely. Thank you.
Thank you, Mahesh.